Hello friends, very warm welcome. Session today we're talking about the apoptosis. What is the apoptosis? Apoptosis is simply a process, or you can say a cell programmed cell death in a sequencing manner. Like one by one, a series is happening in the apoptosis process. So also the programmed cell death is a process in which individual cells do what you can say individual cells die for the good of the organism. For example, in the course of normal development, the course of normal embryonic development, and the organism conserves the cell's molecular components like amino acids, peptides, and so forth. So this is the what? This is the apoptosis. When you have to have embryonic development. Initially, I told you in embryonic development. So, in the embryonic development, you have a structure looking like this manner, looking like this manner. So, this is the limbs of the human or mouse, or you can say any vertebrates. So, this is the organism. So, what is the requirement to apoptosis? The requirement is that the apoptosis cell a particular cells die to make the limbs exactly looking like in this manner. So as a result, we found that this is the series in which we form the limbs in our hand during the development, or you can say in embryonic development. Here, here cells die. Here cells die. In this region, cells die and become more appropriate to looking like this. You can imagine, friends, if your hand looking like this, how this is to be looking something different or inappropriate in that manner. But this is the appropriate manner where we have to count our fingers like 1, 2, 3, 4, 5. This is the 5 fingers in your hand, and some people also may have 6 fingers instead of 5s. So this is the embryonic development, and during the embryonic development, you saw you show the apoptosis process. Apoptosis process. Apoptosis is a Greek word meaning that falling off, falling off, like a falling off a tree, of a leaves from a tree, falling off leaf from a tree. This is the apoptosis, a programmed cell death which is happening at the molecular as well as in cellular level. So, what is the requirement for that particular process in the particular organism? We have some consequences behind that particular cell death. More commonly, you have to say it is a cell death. It is a cell death, or you can say more probably it is a programmed cell death. So, all pupils may have required the growth, or you can say all individuals development process development process. So you can note down that particular point which is mentioned in this slide, and maintenance of the multicellular organism, and maintenance of multicellular. Remember, multicellular, not the unicellular. So, apoptosis happening where? In multicellular organisms, which is depending on not only the production of cells but also the maintenance, the mechanism to destroy them. So here we have the production as well as the destruction, or more appropriately cell die. So cell is produced in a chemical pathway, or you can say by via mitosis or meiosis in multicellular organism, and destroy via the TC, programmed cell death, or more commonly apoptosis. These are the some points why we require the apoptosis, for the growth, for the development, for the maintenance of the multicellular organism, and for the production and the destroy. These are the some points where you can find out the apoptosis process in more appropriately. So friends, we have a structure like this. So what is the requirement for that particular process? During the development, you also know that careful orchestrated means in a series of a life pattern of cell death, help determining the shape and size of limbs and other tissues, which will looking in the previous slide. So friends, cells also die when they become damaged or infected, ensuring that they are removed before they threaten the health of the organisms. So if the cell, if the cell, so this is a cell which is infected, or more commonly viral infected. So what is the appropriate prevention to this cell to prevent another cells infecting via this cell? So we have some mechanism which is apoptosis. So if the cell is die, its neighboring cell is preventing from that infection, from that particular infection. So in the apoptosis process, the infected cell, or you can say damaged cell, also enter into the pathway of the apoptosis. The apoptosis. So if we're talking about the some mechanism which is involved in the apoptosis process, so we have the some initiator caspases. Now, what is the caspases which will discussing about some later. So, initiator caspases, like some caspases are the proteases which will initiate the apoptosis. So, initiator caspase initiate the, or we can say begins the apoptosis. So, we have the caspases 8 and 9. So, in the caspases, what is happening? In the caspase, binding domain present and the protease domain is present. So, this is the inactive monomer. So, when the caspases, initiator caspase, talking about the initiator caspase, is inactive, that means inactive in monomeric form. And when the apoptotic signals induces, or you can say apoptotic signals coming in that particular caspases, so adapter proteins entering, and the dimerization, or you can say dimerization, or you can say activation of the caspases. So dimerization, activation and cleavage. So caspases are proteases which is dimerized to getting active form and then cleaves. So this is the what? This is the This is what? This is the adapter signal. This is apoptotic signal. This is all is the apoptotic signal, which is interacting with the, it is interacting with the large subunit. It is interacting with the large subunit, and this is the small subunit. So as a result, what is happening? The active caspase is present here. So when the caspase is dimerized, it is now activate. Dimerized, it is now activate. So when the caspase is activate, it is initiate, or you can say it is triggers the executioner caspase, which is caspase 367, activation by cleavage. So some cleavage is happening via the caspases to active the executioner caspases. So active caspase, what? Active caspase, cleavage of multiple substrates. So when once a executioner caspase is active, it is now, or you can say more appropriately said, that the apoptotic pathway is running on that particular cell, running on that particular cell. So these are the some mechanisms, some machineries which will participating in the apoptosis process. So in these and most other caspases, which is cell death, not a random process but occurs by a program sequence or a molecular events in which the cell systematically destroys itself from within and then eaten by other cells, and which is eaten by the other cell. More commonly, macrophages are eaten this cells which is destroyed itself, and which is the programmed cell death. So in contrast to apoptosis, in animal cells that die in response to an acute insult, or you can say insult, such as trauma or attack or blood supply, usually do so by a program process called the cell necrosis. Just opposite to the apoptosis. When the cells respond to trauma or attack or blood supply or lack of blood supply, it is going to enter into the pathway of necrosis. In the necrotic cell, cells swell and burst, spilling their content over their neighboring and eliciting in the and influence another cells. So in most cases, necrosis is likely to be caused by the energy depletion, which leads to metabolic defect and loss of ionic gradient that normally exist across a cell membrane. In case of apoptosis, these are the some consequences which is not happening in the apoptotic cell. So which is not happening, like necrosis is what मैंने बोला, मैंने क्या बताया आपको, necrosis is what? It is a cell burst and cell swelling, and also the energy depletion in case of necrosis. So here we have some proteins which is involved in the necrosis, and the DNA fragmentation is happening in the necrosis process, or you can say DNA breaking down taking place in a particular cell which is entering into the apoptotic pathway. So we have protein. This is the protein which is CAD protein, and this is now inactive because of the ICAD. It's inhibitor. ICAD is what? It's inhibitor. It's inhibitor. So it is now inactive when executioner caspases. Remember, you have to know what is executioner caspase and what is the initiator caspase. Initiator caspases, once active, it triggers the activation of the executioner caspases. So this is the executioner caspases which is entering into the apoptotic pathway by the influence of the apoptotic signal. So now when executioner caspase is coming, this caspase normally is to the protease. Cleave the I key, cleave the I key. This is the inhibitor. When the inhibitor is cleaved out, so now the CAD is active now. Now the CAD is active. And when the CAD is active, it is fragmentize the DNA fragment. So this is the fragmentation of the DNA. So which is the molecule which is required to fragmentize the DNA is the CAD molecule. So if the CAD molecule is fragmentize the DNA molecule, we can see on the gel electrophoresis. So this is the time, and this is the, according to time, there is the migration of the DNA molecule is happening. So particularly where the DNA fragment is occurring. Like this is the limb of like mouse. This is the limb. So there is presence of what? There is presence of cells, and this cells, you also know friends, in the cell we have know that there is a presence of DNA molecule. Chromosome is present in the DNA molecule. So DNA molecule is also getting fragmentized when the apoptotic pathway is happening in the particular cell. So this is the region where the DNA getting fragmentized in a particular, or you can say molecular, by the molecular event. So we have two things. And what is the two things? When we're talking about the apoptosis. So apoptosis can be studied in multiple pathways, like the intrinsic pathway, intrinsic pathway, in which the mitochondria, is involved. And if we entering the extrinsic pathway, in the extrinsic pathway, extracellular signaling is important. Extracellular signaling molecule playing important role in the extrinsic pathway. So, and another also which is a granzyme pathway, which is only operating when the cell getting infected with virus, or you can say via virus. These are some apoptotic pathways which is entering into the apoptosis. So in this way, we have to knowing what is the type of apoptosis and which type of apoptosis is. So we have this is the extrinsic pathway. This is the extrinsic pathway. In the extrinsic pathway, this is the killer lymphocyte. So you also know the cell type, immunity, and what is the adaptive immunity, what is the innate immunity, and what is the lymphocytes, lymphatic systems. So that will help you to understanding the mechanism of that particular cells in the apoptosis pathway. So this is the killer lymphocyte, and which is constituting a Fas ligand. Fas is the, so this is the target cell which is going to entering into the apoptosis. When the Fas ligand interacting with the target cell, it's going into the apoptotic target. So this is the Fas, or you can say death receptor, This whole complex is interacting with this region. So the Fas receptor consisting FADD adapter protein, the death domain, this is the death effector domain. So consisting two domain, and together it is called it as a FADD adapter protein. When the death effector domain, this is the death effector domain. This what? This is the death effector domain. So the death effector domains which is present in the cytosol of that target cell. Caspase 8 is coming. Or now what is a caspase 8? Caspase 8 is a initiator caspase. You have to know what is the caspase. First of all, you have to know what is a caspase. So caspase is nothing. Caspase is nothing. It is a protease. It is a protease. Protease. C for cysteine and ASP for the aspartic acid and ASE for the enzyme. Caspase is a protease enzyme. In its active site, cysteine is present and it is cleaved at the aspartic acid residue. So that's why it is called as a caspase protease. So caspase 8 is an initiator caspase. Assembly of disc. Now this is forming to the disc formation. This is the disc formation. The disc with the help of initiator caspase. This is the initiator caspase. So this whole complex is getting interacting with the Fas receptor, or you can say Fas death receptor. And now this is the formation of the disc formation. Now this is the disc formation. This is the nucleus. So activated caspase. Now you know friends, caspase initiate caspase when they getting dimerized, they getting activate. In its monomeric form, it is now inactive. When they getting dimerized, it is active. So this is activated. Why? Because it is dimerized. Because it is dimerized. So the activated caspases, what is done? Activated caspase is done the activation of the executioner caspases. Now the executioner caspases, what is executioner caspase? 367 is the execution caspases, which will initiate the apoptotic pathway of the target cell. This is the apoptotic target cell. This is the apoptotic. This cell is looking like this after the apoptosis. And now this is the Fas ligand is now free. Fas ligand is now free. So entering into the another Fas death receptor, contacting with another Fas death receptor. This is the extrinsic pathway of apoptosis activated through Fas death receptor. This is what? This is the extrinsic pathway. You know what is the extrinsic pathway and what is the machineries involved in the extrinsic path. This is the killer lymphocyte interacting with the target cell. So we have the death domain, death effector domain, and this is together known as a apoptotic adaptive domain. Death effector domain coming in, and caspase 8 is coming and dimerized and the formation of the disc. And this when the disc is formed, this is fueled by the activated caspase 8 and the activation of the executioner pathway. And when the executioner pathway is activating, it is apoptotize the target cell. So this is what? This is the apoptosis pathway. After we have to discussing about the intrinsic path, which is more common, in apoptosis. So what is the intrinsic pathway? This is what? This is the intrinsic pathway. This is the intrinsic pathway. What? This is the intrinsic pathway. I uploaded already a session with that particular at the molecular level. So we have to set that when the release of the cytochrome C, if the cytochrome C is present in the cytosol, it is harmful. It is harmful. You can say a signal for the particular targeting cell to going into or to entering into the apoptotic cell. So this is the mitochondrium. So this is the mitochondria. So cytochrome C is located onto the inner membrane, onto the inner membrane as a peripheral protein, So, or you can say it is present in the intrin, intrin, membrane space. So when that cytochrome C is released from that particular mitochondria, and what is the factor which is responsible to release the cytochrome C? I have talked, I have telling, or you can say I have told you in the previous lecture, in which we have talking about the some factor, some activated protein, some anti-apoptotic, pro-apoptotic proteins. So there are multiple factors which is responsible to release out of the cytochrome C in the cytochrome. So when the cytochrome C is coming into the cytosol, we have some mechanism which is initiate the apoptotic pathway, like the activation of the APA F1 by the cytochrome C. When the cytochrome C is releasing into the cytochrome, or you can say into the cytosol. So this is the APA F1. So this is the APA F1 which is already present in the cytosol. So this is the card domain. This is the card domain. So when the APA F1 interact with the cytochrome C, so this is making a assembly of the apoptosome. So there are 7 APA F1 domains are present and making a apoptosome. So this is the card domain. This is the card domain. This is making the cytochrome and the APA F1 making the apoptosome complex. Into that after that you can say the card and caspase 9 is coming. Recruitment of caspases of 9. So this is caspases now entering into that apoptosome complex and activation of caspase 9 which cleaves and thereby activate the execution caspase. So you have to know what is the executioner and what is the initiator. As you all know friends, initiator caspases. I am written initiator caspases like 8, 9, executioner like 3, 6, 7. So in the intrinsic pathway, there is initiator caspase which is caspase 9 and in the extrinsic pathway. Caspase 8 is present in the extrinsic pathway as an initiator caspase. And finally, what is done? It is done by the initiator caspase to activate the executional caspase. And once the executional caspase is activating, the target cell is definitely going to the into the apoptosis pathway, and that will die. That will die. So this is the intrinsic and the extrinsic pathway of the apoptosis. In next lecture, we will talking about the some mechanism which will initiate the apoptosis process and which will influence the apoptosis process into the mammalian cells. So, and what is the genes? What is the genes which is down regulate the apoptosis? So, friends, I have to requesting to all you that you have to watch my apoptotic video which is uploaded one previously, in which I am talking about the molecular level of genetics. So molecular and cellular, those are the important phenomena to down regulate the apoptosis process. So on particular video, we have to talking about the some activator, some effector genes, or you can say proteins, and some promoting and some inhibiting the apoptosis in particular, or you can say target cell. So friends, I hope you like this session, and if you have any confusion related to this topic, use the comment box, and in the comment box do have cross questioning with. Thank you friends.